So welcome everybody. I'm glad to see everybody's smiling faces here this evening. And if you are shy or in the witness protection program or just really just don't want to have your video on, please do cover and do shut it off because we are recording the meeting. This evening it is my just absolute pleasure to be able to invite and have Dr. Edward Kim from the City of Hope here with us this evening. And as I'm meeting people, I'm going to be double testing. I guess let's see. Let me see if I can get the official title right. Physician in Chief, City of Hope, Orange County, Vice Physician in Chief, City of Hope. Did I miss anything, Dr. Kim?
That's plenty.
Okay, well, it's plenty. It was really hard to get on your schedule because you're really become the man out there as Orange County's really kind of kicked up. And so I am super thrilled that you're here and you're able to kind of go through a lot of what's going on in the lung cancer field and particularly go over the ASCO highlights. And I believe, if my memory serves, you are the first physician, first doctor we've had speak with us since Ceratisid actually was totally FDA stamped and approved. And so we're totally thrilled to have you here and looking forward to getting, being able to pick your mind and ask a couple questions if that's okay.
Yeah, no, I don't think anybody knows everything, so I will share what I can, provide insights with where I can, but, this is a rapidly evolving area in so many different ways, and it's something that is critically needed, and so it's really nice to see the science moving. It's nice to see the FDA not sit on drugs for decades like they used to. So yeah, just keep it. Anything I can do to talk through. It can be on any topic that you want to cover. So I also have a good knowledge in sports and some other hobbies. So.
Okay, great. Well, when it comes to getting time for my fantasy football draft, I will be circling back with you to make sure I get the inside scoop on that. See what you know out there.
I do really well helping other people with that, but if it's my own team, I do really poorly because I overthink it.
Well, okay, I totally understand, and it totally depends on the type of league it is and everything else too, no doubt, right? Okay. Well, all right. So let's start off at the top. What did you see was really exciting and of interest at ASCO this year?
Wow, what a loaded question that is, man. There's ASCO is such an out-of-body experience the last couple of years because of the virtualness. And it's funny, people would ask me, well, which sessions do you attend? I'm like, well, I don't really attend sessions. I go there, I meet with folks, I network with folks, we meet with companies so that we can plot and plan how we're going to do the next round of development, what types of biomarkers, etc. And in lung cancer, you kind of already have hit a lot of the highlights even before ASCO is started. Because you have these in-person meetings, et cetera, and that has totally changed. And I think it's taken a little bit of excitement out of the meeting. So hopefully it can return to in-person in a safe way, in some way, shape, or form, because I do think that we need human contact. But obviously, we want to be very safe. I love the fact that in lung cancer, we are now finally starting to see the earlier stages get more involved. And, when I was a fellow, 20 years ago, seems like a long time now, I had a slide I remember I made, and I was jealous of breast cancer because here I was young, fresh out of training, doing lung cancer and head and neck cancer. And all my colleagues were asking me, why are you doing those? Those are terrible, and I'm like, look, we got work to do. That's what you want to have a challenge that way. And, everybody's like, well, breast cancer is so much better. And I'm like, well, we'll see. And I would say that lung cancer as a field has become the model for precision medicine. Now, you might have HER2 and ER/PR way back when, but there was a long period of time between the next biomarker and those biomarkers. Right. And, finally they got PD-L1 and now they have PIK3CA. But I remember the breast pathologist had no idea how to measure PD-L1. And they had to figure out how to process and send out the samples to get that tested right away. So, but if you think about early stage breast cancer, you have a locally advanced treatment. So it could be surgery or radiation. And then you get a period of time where you're on a chemotherapy or a biologic therapy. And again, I'm generalizing this. And then you go on an oral therapy based on what your markers say. And you take that pill for 3 years, 5 years, 10 years. There's been a lot of debate on length and type, whether it's an AI versus not. But, tamoxifen was a drug that was for the metastatic setting. That's when it first came out. And it was to treat and prevent contralateral breast cancer from occurring. So, people look at it as, oh, tamoxifen is not really a therapy. Yes, it was a pill. It was absolutely a therapy. And because of its profile and its efficacy, it was moved to the earlier stage setting and showed benefit. This is the model, right? So we now in lung cancer have a situation where we're testing for markers. We're using biologic therapies now that supplement our surgeries or radiations. I think we're seeing less and less chemotherapy, which is a good thing. And that paradigm was shown multiple studies, both in the adjuvant and the neoadjuvant at ASCO. And it started with EGFR, but now we're going to see different markers like ALK and others where, why wouldn't you use a biologic therapy to maximize a response and then continue to take one as a adjuvant or a preventative, however we want to label that. This was a lot of the research I was doing at M.B.A. Anderson was, we would call that, there's primary, secondary, and tertiary prevention. And primary is, eating a good diet, etc. Secondary is you have high risk, something like a pre-cancerous area, like a skin cancer that won't, hopefully doesn't progress. And so you do things to try and reduce that. And then tertiary is you've had a cancer, you've gotten rid of it, but you're at risk for another cancer. So you try to do things to keep that away. And, that's what we're seeing, which is great. That's number one. Number two, a little bit more of a disappointing aspect. And this was a large network hospital across the country in US Oncology. And they showed that these biomarker testing rates were not really good. And this is generally a pretty sophisticated group of physicians. It was a little embarrassing. They didn't want to report it at first, but I'm glad they did. The 70s across the board of individual markers. And then on the whole, it was a little bit more like, low 90s, high 80s. And those are the numbers we generally hear. And there's two parts to that. One is the actual testing, but more importantly, the actual utilization. They're finding more and more that providers are ordering the test, but they don't all come back at the same time. So you get PD-L1 back in a few days, you see it's elevated, they're going to get immunotherapy. And that's particularly bad if you have an EGFR mutation present, because that's a situation where you now will harm the patient by giving them an immunotherapy first if they have an EGFR mutation. So that is something that is really coming to light. I think it's got to be a reality check. One could argue that we're behind in breast because of that, but the process in breast cancer with those markers were very inexpensive, number one. They were very easy to do, number two, because the pathologist could run them just right there. And so it was a reflex. I think breast cancer and several other cancers are going to understand the complexities now as more markers are coming into their clinical realm, that you just can't, it's not a single kitchen that can just run everything and get your results out at the same time. If you're you're cooking a turkey, you know, on Thanksgiving, you've got, you know, stuffing, turkey, pie, you know, veggies, and you can't just put them all in the oven at the same time and they're all going to come out at the same time. There are certain ones that get done quicker and others. And so this is the complexity. And we've had that, both that benefit because we've had so many markers with drugs approved in lung cancer so quickly. On the flip side, it has added to that complexity, and people will fall behind very quickly when you don't have uniform, consistent processes across the board.
So as a patient, what should we be?
Very quickly.
Okay, so as a patient, what should we like take away
patient,
and know and look for? Yeah.
Yeah, that's really important these days. I really encourage patients or their caregivers or their family members to really take control and charge. That's not the way everybody believes and behaves, and that's okay. Everyone has a different personality, but you know, being informed, understanding that you need to have some sort of genomic testing done. It could be tissue, it could be blood, and you don't need 500 markers. You know, that the 500 are not going to help you right away, especially at the initial diagnosis. You need the 9 or 10 that we have right now. It's weird that we're getting into double digits, but that's where it's at. And you need that information before a treatment should start, knowing that that could take an extra 10 to 14 days to get those results back. You might get some results back in 3 days, but that's just part of the story. You know, it's sort of like if you are being staged and you could get a chest x-ray, gosh, right away. But we don't make the basis on the chest x-ray. And it takes longer to get a PET scan, but that's a very important test. You need to wait for it. So that's the same thing with biomarkers. You got to wait for the whole thing. And then when you get the whole thing, that's when you can go forward and be tested. Okay, well, I don't know. So that's what I think is very important. Normally, if the wheels we have a straight line somewhere that is we don't want to hear that conversation necessarily.
Okay. Yeah, I'm not sure. Somebody needs to mute.
Well, what he does is. No, he's not written. No, he's got his daddy. Hey, let me see you there. Oh, let me see. Oh, man. Look.
Okay. Hello.
All right, we muted it.
We got it. All right, we got it. All righty, cool. All right. Sorry about that little distraction there.
No, it was sounding very interesting, frankly.
I don't know. It was just getting juicy.
So yeah, that is really important. And I think of the biomarker evaluation, at least the key biomarkers that really are part of the staging and diagnosis, right? And that's how you have to think about those. It's not the additional. That's like saying to a patient with breast cancer, oh yeah, we can start you on therapy. We don't need the HER2 or the ER/PR yet. That means you've totally you could potentially give the wrong therapy. And that is the type of accountability, the type of attitude we need in lung cancer as well. And I've mentioned this multiple times as I've been on meetings now, as we've talked about early stage lung cancer and the treatment. And I said, look, and people complain, oh, well, how come it was 3 years? That was one of the comments I've heard with ADAURA, which is a trial that looked at osimertinib, an EGFR drug that showed a huge disease-free survival benefit. We're still going to wait for the overall survival benefit. It will take some time. But I said, you understand that in patients who have biomarkers with breast cancer, they take pills for 5 years or longer. You know, let's not be so myopic and just think about our narrow little lung cancer world. We have to think about that, look, let's not just criticize a company for the sake of criticizing a company, because that's what people like to do. I know it's shocking to hear that, that thought leaders might do that. But, you know, this 3 years in an adjuvant maintenance setting, to me, you know, is a reasonable number If people want to stop, it's okay. You know, we don't know if what happens stop early or late. But one of the biggest questions that came up, I remember early on in the tamoxifen study, which was stopped early, was that what would have the effect been if people kept taking it for the 5 years or whatever it was intended to, instead of stopping the trial early, and people stopped after 3 or 4 years, they unblinded the other arm and such. So there were questions lingering about that. So I think it's phenomenal. You know, as you mentioned, we have sotorasib now approved. That's great. That's the first step. Now it's going to unlock an entirely, you know, sort of high area of interest. Now there's going to be more discussion around, okay, now what happens when resistance occurs, and what markers appear, and what can you pair this up with, either of these drugs, to for combinations to be more effective. That's the beauty of it, because once you get that approved drug out there, now more people can use it, more people can test with it, and the FDA isn't going to scrutinize you now because you've got 2 experimental drugs in combination. So it's the same phenomenon as what happened with immunotherapy. You look at how many osimertinib combination trials are out there right now. This is what's going to happen. So I'm very excited from those aspects that it will create an awareness. It will create another next level interest in studying, and as opposed to, you know, NTRK, MET-exon 14, KRAS is a more common marker. Not every subtype, you know, but but even those smaller subtypes are on par with, or even more than ROS1 and others. So it is, I think, something that I'm excited about. It's a long time coming. People were always very pessimistic about KRAS, and I'm like, why? Why are we pessimistic about anything? If we haven't tested it, and if we haven't tried, why are we? You know, I mean, you know, we're on Mars now, right? Do you remember when the first Mars rover landed and they said the battery life was going to be 30 days? So we got to get as much information as we can. What was it, like a year, like 400 days the battery lasted? It's like, I'm glad, you know, it's good that, you know, we miscalculated on the wrong direction, because it would have been really depressing if we said it was a year and it was only 30 days. On the flip side, that was great. And next thing we know, China's on Mars now too. So, you know, there was a lot of press behind that, but we're not the only country there. So there's, you know, there is no reason why we cannot continue to expand our universe, explore it, and find something. And, you know, KRAS is just one example. So I think it's great.
For sure. Now, what do you think is going to take for, like, sotorasib, which is now, like, approved for second-line therapy? Okay, what do you think is going to take for it to, like, you know, obviously we need to see trials and we need to do comparative arms and whatever. How long do you think that's going to be before we can actually see it, maybe potentially first line or something?
Not long. You know, I don't know everything that's being planned in the Amgen pipeline. We do have an Amgen phone here, so you just ask them. But there should. I mean, there should already be a first-line trial. I'm sure there is running. There should already be combinations that they've kind of sketched out on a whiteboard and ready to go. You know, when you're first in, I think that's when you have to be the most aggressive. And you have to stick to your guns, but also want to explore. It is biomarker based. So biomarker discovery, I feel, is really important. Finding what other co-mutations exist. There's a little conflicting data out there about co-mutations that we need to sort out, you know, and I remind people, we're not going to sort it out with 100 samples or more. It takes more than that. We still don't understand immunotherapy at all. And the part of what we cannot measure, and it is probably the more important part, is what is the effect of the individual person and their body and their host response and their thing. You know, we're always looking at it from the one side. Okay, we've got a drug out here and we've already tested the tumor, so it has that marker, but that's not the partnership. The partnership is how your body regulates it, how it responds. And so that's why with immunotherapy, we're missing a lot of information. We don't know how people, their body, their microenvironment reacts to these things. And I wish we had a measure on that. Same thing will happen in any of these settings like KRAS. And so I think sotorasib is a great start. I've compared it to like the crizotinib of ALK, and then as we go along, we might develop next generation drugs and next generation drugs. And that's where I hope it takes us to that. Third generation drug that has very little. That's what I hope for.
Oh, definitely.
So those are a few highlights.
Yeah. Okay. Was there
from
anything
ASCO,
else that kind of like jumped off, in general, whether it was around ASCO or other stuff? And then I'm going to start asking you about some of the other stuff. If people have questions, go ahead and start either thinking about them or putting them in chat. It's kind of always easiest if we see them in chat as they come to you. So what do you think about some of the other stuff that's coming in the pipeline and what have you? And it's not all about targeted therapy, right? I mean, it's not all about, we all would like to take, one pill and be magically cured, but that doesn't look like that's going to happen ever. But at the same time, we do need to kind of be looking for, the rest of us that are not, on second line therapy with G12C, or, it's just not like it's a good match for us for whatever reason.
Yeah, I do like, if you imagine a drug development scheme 10 years ago, every subtype of KRAS would get mixed in, and maybe these drugs don't show the benefit, right? When you clump them all together. And then you don't know what you're sort of throwing out with the bathwater, right? By finding these narrower subsets, and not necessarily just focusing on one or another, but being able to parse out the data and look and see how these subsets work. Now we did define something that is very important. Nobody was talking about G12C, 3 years ago, 4 years ago, right? It was KRAS. And now we're getting, it was KRAS and BRAF. Those were the two, right? They were kind of peanut butter and jelly, right? They went together, and so it came from the colon data and other things. It's a different world. And, so there are subtypes. And so I think that's important, because drug development now can proceed in a little more smart manner. We're not going to throw away drugs, at least just from a generalization. That was the chemotherapy approach. Right? That's why all the chemo data looks so homogenized that we could never figure out what chemo would work best in a person. We see that phenomenon occurring in EGFR, right? We used to just be able to say EGFR. Then it was, deletion 19, LA58R, sensitizing ones. Then there was a T790M, which was the resistance. And now we've got all these myriad of uncommon mutations, including 18s and 20s, that now we're developing drugs specific to those, and even have an approval for a drug combination in 20. So, whether you call it zip coding or what have you, it's becoming smarter. And I think these companies that maybe were small companies that had drugs that were very much in a niche area, they're being snatched up now by the bigger companies. Because there's a realization that, look, I don't, if people make money while putting drugs out there, all the power to them, right? Because all I care about in the end is that we have drugs, we have choices, and they're not breaking the bank of people, But, I really think acquiring these companies and letting them develop these drugs further, letting them combine with other drugs, that's the future. And, one of the big things that I've always been very passionate about has been the types of eligibility criteria that we put on these studies. I do not understand why a phase 1 study has to have so many restrictive criteria, because this is supposed to be a safety study, right? And if you put the healthiest people in that study, everything will look safe, right? I mean, you know, we want to put the representative population into these types of studies. I mean, again, it's a different line of thinking, right? Nobody, you know, wants to have a bad event in the study, but you know, we're not talking about a healthy population here. We're talking about people who are fighting cancer, who have already been through multiple lines of therapy. You know, ask them if they're willing to risk being placed on a phase 1 study or not. I think your risk benefit profile changes a lot when you're coming from the first person view as opposed to the outside person view. And so I truly believe we have to be more inclusive, and that will drive enrollment, right? Phase 1 studies have limited spots. It just loosening up the eligibility criteria to make sure it's scientifically valid, not just based on, oh, well, anyone with a creatinine that's slightly above normal shouldn't be on, but the drug is primarily metabolized in the liver. What does that have to do with anything? You know, we're going to exclude people whose disease have brain metastases. Why? You know, we have radiation for that and other things. Why can't we test it? We've all seen effects where chemotherapy helped out, and now we have these super drugs that penetrate the CSF, which I'm glad companies are showing that data and being rational about it. But, you know, why do we exclude the brain but include the liver? You know, that can be equally tough, and it's still a distant site. So I never understood a lot of the rationale. I'm sure it was some very smart people sitting around a table who decided at these meetings that this is the way it was supposed to be, and then it got cut and pasted and added onto. I mean, there's a median number of 37 eligibility criteria per study. Okay. Now, when a drug's FDA approved, does your doctor go through 37 checklist eligibility criteria to make sure you can take it? I don't think so. If you're telling me that a 17-year-old person had a KRAS G12C lung cancer, they can't take sotorasib? Are you kidding me? I was at the DMV today. Might be why I'm in such a perky mood. Had to get my California license, 3 hours there. The person next to me, in the booth, they were doing the eye test. He was missing every other letter, and they were like, "Oh, that's good. Yeah, you're fine." And it's not a difficult test. I mean, literally, it looks like reading a sign. I was like, wow, okay. See, that's how our eligibility needs to be, right? I mean, we need to be loosened up a little bit, right? Because that's the population.
Well, yeah, and they need to ask us. I mean, that's a lot of it, right? Is we need to be asked, because it's not perfect for everybody. And some people will be like the first people to raise their hand. Okay, for a phase 1 trial, it's like, I feel like I have no other options. And there's other people, it's like, you know what, it's not practical because I can't, whatever, I can't get there or something. So I would love to see, them loosen that up a little bit, but I think in order to do that, you probably have to include more people and that's like all the details are a little bit above my pay grade. But I know you've been a big proponent of like making changes to make it a lot more inclusive as far as the trials are concerned.
The beauty of what we've done, Terry, the beauty, and this is where we've included patient advocates, industry, regulators, is that we have made the changes. So the 6 eligibility criteria were changed on the CTEP protocol template 2 years ago. And the FDA issued guidance documents supporting all of this. We now just published another 6 eligibility criteria recommendations in February, and that will go through the same playbook. It will go to CTEP, see if they will change that. I mean, one of the things we had recently was a washout period. I mean, you all may, you're all very educated on this. Do you know what a washout period is? If you don't know, I'll go through.
Yeah, but I'm going to want you to back up a little bit and tell me what CTEP is and all this other stuff.
Yeah. So CTEP is, the sort of the NCI clinical trials drug evaluation arm department. And they set forth, the guidance and the rules and, review any study that's government funded. So it could be a cooperative group or anything. And they have a protocol template, just like they have an informed consent template that one must use. And so anyone who's going to do a cooperative group study or anything that's going to have a government sort of funding around it needs to use this template, not just one that you have on your shelf, not one from, industry. And the eligibility criteria are already templated in there. Well, they took our recommendations and eliminated and modified 6 of those from our publications. So we were very pleased about that, because that's, anything changing in the government, let me just start there, is a very Herculean task. But we had people from NCI involved, and they were very supportive, and we love that. Now, what kind of changes were there? Let me highlight a few. One was HIV. So in any protocol, you have inclusion and exclusion criteria. So these are just like, things that you have to have characteristic-wise to be included, and things that you have that will kick you out or be excluded. And one of those was always listed in the exclusion criteria. It was exactly worded as history of HIV. Okay. So what does that mean? How do you test that? Well, the study actually doesn't ask you specifically to test someone for HIV. So when you're talking to them, you say, oh yeah, by the way, I had HIV, but I'm not on any antiretrovirals. It's fine. Oh, excluded. Boom, you're gone. No test, nothing. We know that the 5-year survival of people who have HIV is exactly of the normal population. So they live just as long because of the medications and everything. So what is it? Now, I can understand if you exclude someone who has HIV who is being treated with antiretrovirals, and those medications are not, they cannot coexist well with the medication that you're testing. So that's an exclusion based on what medication you're on. Scientifically, that makes sense to me. But to exclude someone because they happen to mention they have HIV and they're fine otherwise is wrong. So that was one example. And so we eliminated that, it's gone. Another example was the illusion I gave before. So what's magic about 18, right? Can anybody tell me what's magic about 18? Does your 18-year-old all of a sudden become an adult at 18? Do they actually, yeah, or do they revert a little further back maybe? I have a 20-year-old, so I can comment there. It's fascinating. It has nothing to do with drinking age. It has nothing to do with driver's license age, because all the states, they have their own sort of rules around that. And then 21 is kind of a magic age, but only in some places. So what is it about 18? That the medical community believes that this is something important scientifically. What is it? Now, have you all bought a pediatric medication like at the convenience store, right? So you go there, and if you look at the dosing on that, it'll actually give you specific dosing based on age. And then what happens when it says after age 12? Consult your doctor. Because there is no specific dosing after age 12. At age 12, you are physiologically an adult as far as your liver or kidneys. And sometimes your mind, just depends. So, again, 12 is physiologically where you metabolize drugs the same way. As an 18-year-old, as a 50-year-old, it begins to slow down once you get, 60s and 70s, but it's the same. So why are we denying all these adolescents who are getting cancer and they can't enroll in an adult trial where these drugs come years faster in the adult world than they do in the pediatric world? It just makes no sense to me. And when I that's always my litmus test. It's very scientific. It just has to make sense to me. And if it doesn't make sense, then I like to pursue an answer to justify why it makes sense so that I can understand that, or why does it still exist? So we changed that. That's on the CTEP protocol template now, age 12. Okay.
That's fantastic, because that was a huge accomplishment. And I just above and beyond making any sort of government change at all, like just pro forma things is pretty miraculous. And that saves lives in the end. I mean, it really does. And, you know, so, and like I said, I know you've been, you know, a big advocate for that, which, you know, we view that in the patient community, and you have that reputation with the advocate community, you know. So it makes a difference. And I know that we need to do more of the same, and there's probably so many other things that need to be switched up and changed and modified, and, you know, like, I mean, it's like, I'm not even sure where this whole like normal, you know, body temperature came about from. You know, I have never run normal, right, my entire life in one, you know, more than one way, I suppose. And at the same time, if that was one of the requirements, there's no way I would ever meet it. So what are you working on next with that? Oh, okay. Who has the question?
Yeah,
I had a question. Sorry, go ahead, Dr. Kim, when you're ready. Can I ask my question?
No, question, fire away.
Oh yeah, so I was just curious,
Yes,
from your
absolutely.
experience with KRAS G12C, have you seen it more prevalent in certain demographics, patient demographics, than others in terms of like ethnicity or background? Is it more prevalent in one over the other from your experience?
I cannot, at least categorize yet. EGFR was something that leaned a lot toward the Asian and
Is it just me, or did Dr. Kim go out?
That was where the early clinical aspect, and not. You're not
Dr.
hearing me?
Kim, no, I think maybe the computer doesn't want to hear your answer. Like, maybe it was like,
oh no.
I'm sorry.
Can you hear me now?
Yes, you can. But I think you just kind of like go in and out.
Oh, wow.
Kind of, sometimes. Way.
You know, it's odd. You know, it's been happening, and and I have like all my bars on my internet. I actually have a Google Nest set up that's right behind me, so it's very odd. But it has happened today. So, the is it now? Are you hearing me?
I mean, we'll hear you for the first couple seconds, and then it like goes out. So,
okay.
Or we can just disregard the question. Like you can just type it. Maybe we could do like a quick.
I'm gonna switch computers real quick. I have two.
Okay.
I know. I'm one of those people.
My question broke your conference. My bad. I'm gonna mute myself now.
Okay, I'm gonna switch computers real quick. We can't have a dialogue if I'm going in. You'll have to let me in, I believe. Okay, I'm trying to log in on the other computer. All right. Boy, it might be my internet. You guys hear me on this side still? Yes? No? Thumbs up? All right, I'm gonna still talk, but give me, it's very, very tough to give answers. All right. Okay.
Terri, if you're talking, you're also on mute. I just see your mouth moving and I don't think any of us can hear you. Sorry. I know I've been watching everyone, so like, I don't know if you needed us to hear you or not. But.
How about now? Can you hear me?
Yes.
Okay. All right. So to answer your question, which broke my computer, by the way, I have not seen a predilection for a specific demographic yet. You know, unlike EGFR mutations, where we knew which patient population was more prevalent even before we knew what a mutation was, right? Asian, female, never smoker, adenocarcinoma. The problem that arose there was that we had preconceived notions based on early subset data of which populations had those mutations. So there were some populations that actually then people weren't testing. And this especially happened in the African American population. There was early small data suggesting that in Hispanic and African American populations that EGFR mutations didn't exist, so you shouldn't test them. So these people were not being tested. Again, based on what? Finally, after 10 years, there was a larger definitive data set that actually showed that the incidence of EGFR mutations in African American patients was the same and maybe even a touch higher. So, you know, I'm glad on the one hand we don't have groups that we've already said, oh yeah, that's a person who's KRAS, because we need to test everyone, and I want everyone tested from that regard. Does that make sense? Did you hear what I said?
Mm-hmm.
Okay. Or you just, I'm glad. Nodding your head still just makes me feel more comfortable, even if you don't hear me. So it's fine.
Thank you for answering that. Appreciate it.
But I will say that it's still going to take some time to get that type of data and see. But right now, I would err on the side that everybody needs to be tested.
Well, yeah, I, you know, from what we see, right? I mean, we're just kind of like all about getting biomarker tested. And at the same time, my personal, the little bird that I will put underneath my saddle, as it were, is that when it comes to all the studies, I see smokers and former smokers all looped into one with a tobacco history. And I don't think that's necessarily accurate, because somebody who just had a cigarette this morning or quit smoking 5 years ago is completely different than somebody who hasn't had any tobacco usage in 30 years. And I don't think that it's necessarily giving us all the information that we could really be gaining from information, from the studies. So, okay, let's see. I saw a question that says, okay, let me read this question here. It says, hi, my mom had her first dose of Pembro this morning at City of Hope. She has PD-L1 of 95% and G12C. She was supposed to start trial, but they lowered the dosage and decided to give her Pembro at cycle 3. We've heard such great things about sotorasib. Do you think adding this drug later on would be possible and effective? Dr. Selja is incredible, by the way.
Yeah, no, he's great. And, you know, the problem was, again, with sotorasib, it wasn't approved as an initial therapy, right? It's after therapy, and I think that will change quickly. To me, you know, if you found someone with a G12 KRAS, you know, G12C mutation, I would be, and they were untreated, I would be very tempted to want to give them the therapy first. You know, the insurance company may not let you, and I don't think anybody should be paying out of pocket for these types of drugs, so we can always use it. And we know we have very good therapy compared to 15 years ago with, you know, chemotherapy and IO. So I think that's always a better, you know, better second choice. But I tend to always want to treat with driver mutations up front. And, you know, whether that's a clinical trial or such, we know what we have with standard therapy. We know it's there. I think it's very rare these days that we lose opportunities of treatment because we watch our patients more closely and we're monitoring them, and the drugs aren't as toxic as they used to be. So I always ask myself at the end of the day, it's like, all right, you know, what would be the most regretful if you didn't get a therapy into somebody, right? And I still have nightmares about a patient who I was treating at M.B.A. Anderson. This was a while back. And he was a young man in his 30s. He had a little bit of a learning disability. So he and his brother were very close. His brother was an investment banker, and they would fly together. And remember, his name was Joey. And so his older brother was always very sensitive because, you know, I was a second opinion type doc. They were seeing someone in Atlanta already. And, you know, Joey understood what I was talking about. He was a little slower and stuff, but he knew. And I dealt with him directly. You know, his bro's there trying to help, but I'm like, no, I got this. I'm talking to Joey. And, you know, we communicated very well. He listened to every word I said. His family would tell me he'd go home and he'd be like, okay, Dr. Kim said all these things, and he's got this notebook and this checklist, and he's doing these things. And he was, you know, young people tend to have more aggressive sort of, you know, cancers sometimes. And this is early 2000s, mid 2000s. So not even close to the era we're talking about right now. And MD Anderson, you know, we were trying to look for things, etc. In the later stages, after I had treated him with several therapies, I resent relapsed and sent tissue, and we found an ALK translocation. Like, holy cow, you know, and this is before crizotinib was being approved or anything. And it was that sort of period between the drug studies were kind of wrapped up and FDA approval hadn't occurred yet. So I'm trying to get this drug. And back in the 2000s earlier, that, you know, they just weren't as liberal with getting these drugs out and stuff. And I remember his disease was progressing. He was in the ICU. He got intubated. He was in Atlanta. And I'm like, he was literally supposed to get, there was a local practice because Anderson couldn't get it. There was a local practice that was going to get the crizotinib for him. And it was supposed to be like this week, but then it got delayed by a week. And that's when he ended up in the ICU. And I'm like, my God, can we crush the thing, put it through the NG tube? What can we do here? You know, I mean, I'm like, my God, you know, and I called Pfizer, asked about formulations, etc., just nothing. And it just couldn't get the drug out quick enough. And so, you know, am I ticked off about that? Yeah. Is it anyone's fault? No. I mean, the drug wasn't approved. It just happened that he got sick in the no man's land period, and there was not any other drug. This was a first in class. So everybody gets really nervous about a first in class drug, and, you know, oh, we don't have any experience with it. You know, you got to be careful. It could be toxic. How well, you know, that's what sotorasib is going to go through, right? But, you know, there aren't that many people who touch this drug, you know, and, you know, the response rate isn't, you know, 60 or 80% on it. So, you know, you might have some people who don't buy into that. But no, I think you want to try and push these drugs that have targets. And I still remember that. So, you know, as you think about this question and, you know, what you would have regrets about, if you've got a driver mutation, you want to make sure, do everything in your power that you get that drug that targets that mutation in you in some way, shape, or form. That doesn't have to be first line. It could be second line, but there needs to be a high level of assurance that this is going to happen, because that would be a regret, in my opinion, of not having the targeted drug to that tumor at least tried. 100% of people do not do well on a targeted drug, and even with EGFR and ALK and others. So remember, that's not the expectation, but at least you're giving it a intelligent and scientifically rational thought behind it. I mean, some of my best responding folks took chemotherapy. I have one woman who's alive almost 20 years with stage 4 lung cancer. She took chemotherapy. There was no targeted therapy back then. I know she didn't have an EGFR mutation. I have no idea why she is alive as long as she is and doing as well as she is. And that's not really, that's okay, because I'm not God and I don't know all those things. So, it just happens sometimes. And you just want to do your best to try and make sure you are giving every advantage you can so that you can try to make something like that happen. But that's not me.
Do you know anything? Can you think of anything that might preclude you from using, a targeted therapy like that? I mean, is there something that just jumps to the top of your mind to be like, oh, this would probably not be a good first choice?
Only if there's a direct contraindication with heart issues or hypersensitivity. A lot of the TKIs don't have that. I've seen some people with explosive rashes and diarrhea that can occur, and that's obviously not a good thing. It's, again, just that hypersensitivity. Cardiac arrhythmias is sometimes that's a no-go, because we don't want to help with one thing and then have you die of an arrhythmia on the other side. So that's not a good thing. Although I do think we overblow QTC prolongation. That is a situation where in a heart rhythm strip, there are a couple of bumps, mountains and valleys. They measure this distance between a hump and a valley. And if it's longer than a certain length, they get worried, because then you can start getting irregular beats. Many of us argue how clinically useful it is and whether it should really stop someone from taking a drug like a TKI, because that is generally one of the things that excludes people. But I'm not a cardiologist, so I'm not going to comment too much on that, but it's just a point of contention. Again, risk benefit, I think you have to do that. Okay,
but anything else you can kind of think of offhand? I'm just asking. I don't know.
Yeah, no. I think of our approved drugs out there. The reason why BRAF hasn't taken off as much as it has is because it's an IV drug formulation. There are quite a bit of side effects with it, and so although we have targeted therapy to BRAF, it's not the kind of targeted therapy we're used to. That is a easier to take oral therapy, etc. There's still a lot of maintenance around BRAF treatment. So that's why, and that's a 3% incidence. That's not, minuscule, but that's why you don't hear a ton about it.
Dr. Kim, I'm sorry, can I ask one more question about, you mentioned
Please
that you had a patient that was on chemo for a very long time and has done really well in stage 4 and didn't have a actionable therapy at the time, right? So would you transition her to a targeted therapy if it became actionable, considering how maybe the safety profile might be less serious? Or do you feel like, my patient's been doing well on chemo, I'm just going to stick with that? And I mean, what has been your experience with that, as all these targetable therapies become actionable?
So I'll answer that question as two parts. One is that my patient was a little unique. I didn't keep her on chemo for 20 years. She actually only took 6 cycles of chemo, and I went a little outside the box and consolidated her disease with some radiation because it was limited at that point. She had brain disease that recurred a couple of times that we were able to use SBRT, but other than her first initial chemo that I gave 6 cycles of, she's had 0 systemic therapy since that time. It's unbelievable. So it just means her immune system had good memory, locked things down, and there it is. Now, to your question of the pragmatism of someone who's on chemotherapy, it's working, it's holding things at bay. The real caveat there is that, there are some docs who really like this maintenance chemo aspect, and that's fine as long as you do not continue to erode the infrastructure underneath. Meaning, I don't want to wear out the bone marrow, right? And by keeping someone on chronic chemo, that's challenging, right? I did have someone who was on Alimta for 3.5 years. But toward the end, I had to dose reduce it, which you don't even need to do with Alimta, but I did. I spaced it out instead of 3 weeks to 4 weeks to 5 weeks to even 6 weeks. And so, and that person didn't have anything actionable. I think if I don't think indefinite chemo, even though it may keep things at bay, I think part of it is also we fool ourselves, right? What would have happened if I had stopped the chemo at any one of these points and observed for 3 months? Would the disease have grown or would it have stayed stable? I don't know, because I don't know how the immune system was affecting that tumor. And nobody wants to risk that because it's working. So we don't stop things that are working. It's like, you think of Las Vegas and at the blackjack table. I don't gamble, but I'm just using observation. That's why people keep going, right? They just keep going, where you should walk away at some point because you're ahead. But this is the tough part of the scenario you just described. So I believe in giving breaks to people. I don't think chemo ever solved the problem, especially a chronic treatment of it. And that if you can then, my suggestion would be treat someone with chemo, it's working, you keep them on a maintenance, it's working. As the side effects start to add up, I stop the chemo, give people a break. They get very scared about it. I'm like, look, we already have a plan B, so I would rather have you ready for the next round of therapy rather than wear you out, stringing you out on chemo. So, it is a marathon. This is not a sprint. And that's how I used to talk to my patients about that. And so if they have a driver mutation, even better, because we already know what the next therapy is, and we will fire it right when we're ready, when the disease is. But if I can allow people to be off therapy and we can watch their disease closely, great.
So,
chemotherapy is not a preventative.
So Dr. Kim, how long, I know this is a difficult question, but how long do you feel someone can stay on Alimta as main?
Very different among the individual person, Nancy. Like I said, the woman I treated in Houston who was on Alimta for 3.5, 4 years, she showed up to my office not being able to walk and had a big brain lesion taken out of her head. So she was kind of in a fog for the first 6 months I was treating her. I don't even think she knew who I was very time. She was very sweet, and she was probably in her late 60s. And it was very interesting. This is a really funny story too. So her family shows up with her all the time, and they're very supportive. They lived out in Katy. And so we're treating, and we're like, wow, she's getting really great responses here. She's actually recovered now. She's walking, she's healthy. And one day she kind of just woke up. You could just tell, and she showed up and none of her family members showed up. And I'm like, this is weird. She had her son, her daughter. So I'm like, okay, maybe they just couldn't make it. Fine, she's fine. We treated her. And then a couple cycles later, like the son shows up. And I'm like, hey, I missed you last time. What's going on? And they were like, well, you want to know the truth, Dr. Kim? I'm like, yeah. She's a really horrible person to spend time with. And we all don't want to spend time with her. But when she was in that fog, she was really sweet. So we all came and were able to spend time with her. But now she's back to her baseline. And she's very cantankerous and very much, it was so funny that this was the situation. And I'm like, I got it. And actually it was true. So many, I had a doctor out closer where she lived who was helping just give her the doses of Alimta, and I was helping manage it. Oh my gosh, she just I think she wanted to fire every one of those docs out there. I'm like, we need them. we need them as part of the team. But yeah, this is like the only one time I can remember something like that happening ever, where family just stopped showing up, and I'm like, why is that? What's going on here? It's not like, she's doing really well. So the answer is that she was able to tolerate it for 3.5, 4 years, but you just want to be mindful that the person who's treating you is watching how those blood counts look. And again, because you just don't want to wear it out. And some people are so resilient, their blood counts never change, and other people are so sensitive. And that's the part that we can't measure, is what someone's infrastructure does and how it reacts. So Nancy, I would say that question is actually very individualized, and that some people don't tolerate 6 months of therapy. Other people can go on it for years.
Okay.
How long do you usually put people on immuno?
How long?
How about immuno? Yeah, how about immuno? If you use the same guidelines?
So, the way we scientifically came up with the immuno guidelines, and it wasn't me, but I mean, I include myself in this scientific, morass, is the trials were originally written for 1 year of treatment. So you got chemotherapy with immuno, and then you continued the immuno, and you get it for a year. People were doing really well after a year. They're like, we're going to stop this drug? Why? I don't feel it. It's fine. Okay, we'll continue it for 2 years. Okay, so then it became 2 years. Then at 2 years, it's like, still feel fine. What should we do? Yeah, I don't know. It's going to cost a lot of money to give another year. We're not sure what the benefit is, so we're going to stop it at 2 years. So again, nothing magical about 2 years, other than awesome that you got there, right? I mean, that's phenomenal. Just like remember that age 18 thing, right? What's the difference between age 18 and 16? And again, remember that other part that I talked about that we can't measure. What is the internal speedometer, or that measurement of each person's own immune system response? And so there are some people, Terri, that I've given 3-4 months of immune therapy to. They had a side effect, had to stop. Well, guess what? They're fine a year later or 2 years later. And I'm positive if I continue to give them the immune therapy, we would say, oh, look, if the immunotherapy is keeping the disease at bay and stable. But in fact, we had to stop because of side effects, and it was still a year later where, so the way it works is it's not the therapy that's killing it. That's what we're used to with chemo. That's why we don't want to take someone off Olympta or something. This is exposing a vulnerability in the tumor. That's what these immune checkpoint inhibitors do. You guys have all watched Star Wars, right? No? Lisa, are you kidding me? Okay, I gotta leave. I'm sorry. Here we go. Wow, Star Wars? Come on. In the age of Netflix and, every possible, I get it. Some people didn't make it to the theaters.
I know. I've been to Disneyland. I love Star Wars, like the land, but I just couldn't get past like episode 4. And I know.
Well, episode 4 is the best one. That was the
I
Luke
know.
Skywalker.
That was
if you
the
watch
first
the
one I
first
watched,
3,
right?
they were horrible. Yeah,
I know, but I try to do it in order. But
okay,
sorry,
well,
I know. I'll try.
It's the same theme on all of them. Is that you have a Death Star of some sort, and you have to take the shield down, and then that's when the little fighters can come in and kill the Death Star. Right? That's the premise. So think of PD-L1 or PD-1, the therapies, the immune checkpoint inhibitors, as going in and deactivating that force shield around the Death Star. Ben Kenobi did that on the original episode 4. That's what happens. Now it's exposed, and the fighters can come in, which would be like the T cells or somebody else. So the tumors have a defense that they've built up, which is the like overexpression of PD-1 or PD-L1, and that's where these immune checkpoint inhibitors do. They don't kill the cell, they expose it, and then your own body comes in and kills the cells. So it's your own body that's fighting it, and the immune checkpoint inhibitor has allowed the tumor to be exposed, take off that cloaking mechanism, so that now your own body can fight it, and it recognizes it as foreign. So that's why, Terri, to the question of how long should you take an immune checkpoint inhibitor for, it just depends on how rigorous your immune system is. And, if it recognizes the tumor right away and can adapt and suppress it, you don't need that much, you don't need years of immune checkpoint inhibitor therapy. But we just don't know. So that's why 2 years and we stop. So I stop at 2 years. If somebody has to stop at 3 months, 6 months, I explain to them exactly what I just told you about, okay, it's not really about the immune checkpoint inhibitor. It's about your own body mounting a defense against the tumor and being able to recognize it. And taking away the PD-L1 recognizes it. Now, what happens is that tumors are smart, so they tend to adapt and they like to mutate. That's why we see these different mutations occur. And that's where it begins to evade your current defense and then mount a different type of attack. So that's what really happens at a recurrence, is that the tumor adapted. It's trying something different. And that's why it's important to rebiopsy, see what those new markers are that are targetable, and then change and switch the therapy as we need to. So it is this kind of chess match between us and the tumor. And that's both sides are trying to be smart about it. And Lisa's got to watch Star Wars.
Yeah, definitely. I think I need to rewatch it. So, okay, but just to clarify, so sotorasib right now has only been approved for second-line therapy after immuno, is that right?
Yeah.
Yes. Okay. And so, like, the question that Mary posted here is, do you think sotorasib should be first line for G12C? You kind of skirted around that pretty delicately. How do you want to answer?
No, I'm very direct. I think it should be. I think any driver mutation therapy should be the first therapy. And so I think it will follow shortly. Again, I don't want people paying, tens of thousands of dollars just to do that. Immune checkpoint therapy is good therapy right now. So it is much better than the traditional just platinum doublets that we had before. So that's why my uncomfortable level is less than it was 15 years ago. Right. Okay. So, but I still prefer, if given the choice, and if we can do it, to start with a driver mutation.
Okay. And I know that as data starts coming in and whatever, there's going to be a lot more, it will be a lot more nuanced, I guess. A lot more nuanced. Yeah,
it's a good thing. I think, I always think more to the party is better. And so, I hope there's 3, KRAS drugs out there in the next 6 months, and that, we have to debate, when, where, which one. That's great. I love it. Let there be choices. And that's what we're all about.
Oh, definitely. And as we know, because we last month we had Dr. Awad on, and he kind of talked about the resistance to the upcoming inhibitors and what have you. So we know it's coming, and it's coming down the pike,
Yes.
So at least we're getting a little bit more prepared for it, I suppose.
I think it's great.
There's yeah.
I want it. Bring it. I want more. It's a great.
Oh yeah.
It's a first word problem.
We're cheering for it. I assure you, every step of the way, and not just for G12C, for and not just in lung cancer, but across all the KRASs, right? And there's an awful lot of us out there that still don't have any sort of targeted therapy available, and hopefully we'll be able to find out what's crossing and how come it works for lung, not colorectal, and we might be able to, cross over and figure out, like with G12D towards pancreatic or something too. Maybe there's so much involved in I don't know. It still feels magical to me behind the scenes what goes on in order to be able to acquire all the data and then come up with plans. So
yeah,
I'm just going to throw it out there. Does anybody have any questions out there?
I saw one that came through from Mary. She asked about the rebiopsy. And so the preference, Mary, is any time that when a therapy stops working, which would by definition mean that the disease has changed. That could either be growth of an existing disease area or a new area that has become affected. That's when I like to do a biopsy. A tissue biopsy would be great, but you also have to consider safety reasons as well. Liquid biopsies are very nice, non-invasive ways to get the information about the genomics. So I know many colleagues who are using that. If I can use a tissue biopsy and do it safely, that's my first preference still. Because you want to establish if it's a related cancer, it could be something different. So that's what I do. There are many places, Lisa, that send both liquid and tissue. We were in a pattern where we would use Tempus where I was before. And the reason why for Tempus is because you actually do collect the blood at the same time. What they do is that they run the tissue, and if you get a insufficient quantity or something, then they reflex and run the blood. And so you could get both from that regard. I think you will see that trend more and more these days.
Okay, does anybody have any questions? They just kind of like unmute yourself. It's a small group this month. It's probably because everybody's on vacation and we could travel
Good.
for the first time, right?
Good for them. That's what I would say.
Yeah, no kidding. No kidding. I think we all need a vacation from life for a little bit here. Does anybody have any questions they want to like shout out? Okay, I see. Just thank you. Okay, 17 months, Mary. Wow, that's fantastic. That's fantastic.
What's that? Let me see. Oh wow! Congrats. That's good.
Okay, all right.
That's real good.
I have a question. I have a question for you. I can come up with something. All right, so we got.
Don't let Terri ask questions. Someone else ask a
No,
question.
don't let Terri ask questions. She'll just derail the whole thing, right? Next thing you know, we'll hit up my fantasy football league, and then we start like worrying about all that too. A lot of because we do get international patients in here, and we'll see the mutations written differently. Can you kind of like explain to me how that we might see them reflected differently rather than just KRAS G12C or something?
Well, I think what we were seeing in EGFR early on was that they were just measuring, they would just label it as EGFR and then just, list out some drugs and a few of the, and not be specific about the individual mutation. And so it'd say like present. Like, what does that mean, right? There are a lot of EGFR mutations that are present. So I think we see that less now, especially in the US with the commercial tests, but that was an issue before. And you never know what smaller labs, because there's still a lot of local labs doing this type of testing that have affiliations with hospitals. And that's part of the reason why I believe testing is not as high as it should be, because some folks who are just doing it purely out of whether it's a good business model or not. And, that's tough. So no, I think that you definitely want to have more detail in that. But, there are some clinicians who also don't want the detail because it is too much detail. And when you think about yourself being in a more rural area or not having a resident expert down the road, I mean, all these academic centers, we can always say, oh yeah, there's somebody who can help explain this. You don't want to be that person who's calling the 1-800 number, 1-800 Foundation 1, please, please explain these results to me. And that's hard. I empathize with those community oncologists out there who have to, see one lung cancer patient, then a colon patient, then a breast cancer patient, then a lymphoma. I mean, that's a lot of stuff to keep track of in 4 patients. Whereas, as experts, we're seeing the same thing all day. There's variations in that, but it still narrows the field. So I think that's something that you have to be very cognizant about. The other question on here about toxicity or negative effects of adding sotorasib after pembro, so sequencing that way. No, I mean, the studies were done that way. Certainly taking pembro or an immune checkpoint inhibitor first and then sotorasib later, sequencing it is certainly fine. There are some combination studies that are being done. I don't know what those toxicity profiles look like, but that's something where I don't think it needs to be added to right now. You want to let pembro have its shot and see if it's working, and if it's not, then you have the next step as far as a treatment.
Anybody have any questions? Unmute yourself or
Hi.
chat them in the chat. Go for it.
Hi, thanks. That was my question, actually. So I wrote up before, we were actually going to start the Amgen trial this morning. We got approved last night. And what they were supposed to do was the pembro first and then the sotorasib. But instead, Amgen, randomized and lowered the sotorasib to 120. And at first, Dr. Salge was like, okay. But then he came back and said he had just spoke to the head of the trial, and they both agreed that it makes sense to do the pembro first, and that there was some toxicity seen with the combo. So we were just a little confused and taken aback because we were all like, okay, we're ready to go. And we've heard this sotorasib is like a miracle. And then he was worried the mass could get bigger and that it was kind of too late to wait till cycle 3 for the pembro. So I just, like you said, I don't want to miss out kind of on the sotorasib. And he said the trial's always there if we want to go back to it. but I just didn't understand really which should be given first.
Yeah. Well, following the letter of the law of the way the FDA has it, it's still immune checkpoint first and then sotorasib.
Right.
And I think why there would be sort of some hesitation with a lower dose is that, again, sotorasib didn't have a response rate that was 75%, 80%,
Gotcha.
right? And so, it was fine. It's been a point of criticism with some folks that, having a, response rate in the, 30 to 40% range, what does that really mean it's targeted? Well, it did extend a lot of things. And so that's why I compared it sort of to an initial sort of crizotinib type situation, right? But I don't think, first of all, you look very well. And, something.
It's my mom, but thank you.
Okay. All right.
But she looks very well too. I mean,
okay. If your mom looks well, the worst thing that
I'm the mother.
Oh, hey, hello.
Hey, Mom.
Okay, I'm gonna have her go walk outside now and sort of do a few circles. No, I'm kidding. Stay in the car.
Her physical has been perfect. You wouldn't even know, but unfortunately, that's the situation. So yeah.
So what's the worst case scenario? I always think of
so
go ahead.
I just want to tell you another thing is that this is her first line of treatment. We found out on a CT that was done at the end of April, but it was probably missed last year. That's why we're actually we live in Las Vegas now, but we've been traveling to City of Hope every week just for you guys because everyone's been so incredible. And obviously they missed it in Las Vegas. So with no treatment for so long too, he was really worried that, we have to wait the 6 weeks when her PDL1 is 95%. And so I totally understood that. I just kind of, like I said, luckily you guys are talking today and I figured, let me just ask again.
Yeah, no, what's nice is that, so if you look at the sotorasib response, right? And we don't know if it could be higher in untreated folks versus after immunotherapy.
Gotcha.
And you look at even the single agent response of Pembro in a 90+%, it's in the mid 40s response rates. So I always look at things as like the best case scenario and the worst case scenario, right? Because those are the edges that we live on, right? The best case scenario is Pembro works incredibly well. It shrinks the tumor and, your mother doesn't feel a lot from it. The worst case scenario is that she gets an immune side effect that completely knocks her down. And, again, that's a very rare thing. It happens much less in patients with lung cancer than melanoma and some of the others. I mean, we don't know why. Side effect profiles, you would think they would be pretty similar if it's the same drug. But in fact, it's very interesting. The phenomenon is that immune checkpoint inhibitors are tolerated much better at same doses in patients with lung cancer and others. So yeah, it's very strange. So she looks fine. She sounds like she's had some therapy already, right? She has started the Pembro or not?
My first
this morning was her first, actually at 1 p.m. was her first Pembro. We're on the way back.
Nice.
So far she's okay, but
how's she doing?
So far so good.
Fine.
She feels okay, but they gave her a Zofran too. So
is she glowing at all?
I hope so.
Okay.
No, I never felt sick. I feel totally 100% fine.
Nice.
But I guess if I took chemo, I wouldn't feel fine.
Well, who knows? But,
well, you don't know. Sometimes people feel just fine with chemo too. Again, it's the other side. So.
And we also went in for surgery on June 7th. We were supposed to have a lobectomy, but it had spread to the chest cavity, so that's why we're here.
I gotcha. Well,
yeah.
I think being at City of Hope, number one, is a great move. Las Vegas, although has many different centers there, there's not really a center of excellence there. So I'll just start that. I think Vegas is a great city. There's great people there, but you need a quarterback. Dr. Salgia is a very good quarterback from that regard. Second is that, Pembro is fine. You really will know really in the first couple of days whether someone, is having a side effect, and she looks great right now. There can always be latent side effects that we look for, but, that you can't control that. So there's no reason losing sleep over it, right? It's like no different than I tell people to, you're in a car right now. If I gave you informed consent of everything that could happen from your drive back, and that doesn't matter if it's 5 hours or 30 minutes, that's when reality sets in, right? I mean, people get very stressed out about stuff, and I'm like, look, there's equally number of people dying on the roads as well. Wear your seatbelt, put a bike helmet on, what have you. There's a lot of things. So, we don't get to choose how we depart the earth. So that's the perspective. But no, I'd say, okay, we've chosen Pembro. You ride that horse until it stops working, or if there are signs that your mother is not tolerating it well. And those are the two reasons why we stop a therapy is, excessive side effects or inadequate results. And then you've got sotorasib just waiting for you there. You don't have to be on a trial at that point and just give it to you. So, that prescription gets written and it gets filled very quickly through our specialty pharmacy and you're done.
I really appreciate the information. Thank you.
Thank you.
And we've been telling you the same thing about the car rides and all that. So it was nice to hear it from you as well.
Oh yeah, no, I used to have a person from North Houston who used to drive down to see me. It's about a 45-minute drive. And she was like, well, I really should get all my treatment here, right? I'm like, no, you can get your treatment locally. I'm directing the therapy. Any board certified oncologist can give you the chemo that
Right.
I'm recommending. Like, no, I feel better coming down here. I'm like, okay, well, Houston traffic is not really the greatest thing. And so, you
Like
need
LA.
my brain,
Yeah. Yeah. Just like
you know-
LA, but we're coming as much as we need. I told her, I mean, some of the doctors in Vegas are good. Like I work in OB and the doctors are amazing in OB, but other areas it is not so good. So
Well, if you guys want to mix it up every now and then and, spend a few days on the Newport coast or, in Orange County where it is much nicer, let me tell you, then just say, hey, Dr. Saldia, can we get our next dose down at Newport Beach? That's one of our Orange County clinics. And he'll say yes. And we can do it there. So.
That's a
it's a
great,
great idea. We're going to tell Dr. Salgia that he's having us come back next week already. So that's our next goal, beach one.
It's 72 degrees and cool on the coast here. So I'm sure it's about 120 degrees in Vegas right now. So,
Cool.
Just
All
something to
right.
think
I see. Okay. I see another question. Okay. Is it true if you go from immunotherapy to a clinical trial and develop resistance, you cannot go back to immunotherapy? And is there any data about that?
No, and that's a good question. It's the same question that a lot of scientists are asking themselves. Immunotherapy does, and we thought chemotherapy does this too. It sort of resets the landscape, right? So you develop resistance. Again, think of that chess match, right? And so we start with a tumor that has somehow now evaded your immune system and is growing. We start now with a therapy. Let's say it begins to work. Tumor doesn't like that. Tumor now is trying to figure out how to survive, how to adapt. It develops a mutation, a rogue variant, starts growing. Now we re-biopsy it, try to see what it looks like. If we have something that we can target it, we hit it now. I have reutilized the immunotherapy several times in patients because, and there's actually data out there to show that immunotherapy, even if it stops working, can somehow sensitize or prime things for even chemotherapy to work better. So they showed higher results, response rates with things like navalmine and docetaxel, which are ancient chemotherapies in the heavily pretreated population that had higher response rates. And these are sort of some conglomerated data sets from places like Memorial Sloan Kettering and Boston and others. So we all have that belief. I believe that, radiotherapy also has an agitating effect to help with immunotherapy. So I was always trying to find an excuse to give somebody some spot radiation someplace and then follow that with immunotherapy because I thought that, that's again, that's Ed Kim talking, not NCCN or anything like that. So, just saying is that there's a lot of hypotheses out there that we still want to try and figure out. But that's what makes it exciting in the fact that we have many more things in our toolbox that we can explore and test, rather than does chemo A beat chemo B. I mean, gosh, I just want to put my head through the wall when I see trials like that, you know? So, and then a question about the Mirati compound, the 1133. Yeah, I mean, it's being tested. It's a G12D drug. You'll see other variants as well that will test different compounds. So it never ends. And, you know, that's why the approval is so important. Once you get an approval, now you're just going to see, you know, that's why it seems like we have 2 or 3 drugs in every one of these sort of small little biomarker categories, because it just spurs that innovation. It really goes. So yeah, I think absolutely they have one. I don't know if it's effective yet. We'll wait for the data, but I love the fact that you can get drugs approved based on early phase studies with single cohorts if they show high degrees of effectiveness. That's a big shift from the old FDA of having to go through phase 3, you know, randomized placebo-controlled trials, only looking at survival. So,
Yeah, we're all for that. Yeah, I'm hopeful for many of us. You know,
I always feel like we haven't. I mean, if you think about the number of drugs or indications that have had to be pulled back by the FDA, let's think about that, right? Can you think of that many, even off the top of your head, where something was approved and then later it was pulled back because when we got the bigger results, it didn't show what it should. That is what we have to measure, okay? That is our measuring stick, not the number of drugs approved. It's the number of drugs we pull back. And like with anything, there's going to be times where we miss. And that early data can point us in one direction. Again, it's that risk benefit. I believe if we don't have a certain number of drug approvals pulled back, let's say I'm making it up 5%, that means we're not approving enough drugs quicker. Okay, it's like safety recalls with products and stuff. Yeah, we hate it that a fire started on this battery or whatever, and you know, or a laptop or an iPhone or what have you. But that also means we're not getting enough products out there. That means we would stick with our one, you know, 20-year-old technology that didn't cause fires. And, you know, to drive innovation, to drive technology, to take that next frontier, that's what it needs. That's why astronauts are very brave people, because they're exploring things for the first time. We don't have a track record there. You know, you can't simulate that kind of stuff. That's what I think in cancer therapeutics, it has to be. So again, I always believe we're not measuring the right thing. If we're not failing at things, that means we're not putting our goals high enough and we're not trying enough of those shots, right? You can't be LeBron James or Michael Jordan and miss that last second shot. You know, they've got to take a lot of those shots. That means you're there. You had the opportunity to take the shot. And if you missed all 15 of them, fine. But at least they had the opportunity. But they're not going to. So that's what I believe in drug development, you know, with the FDA and how these approvals occur. You know, but everybody gets hypercritical when there's one drug that gets pulled back and then it's, oh, patients were exposed unnecessarily. And, you know, that's a news reporter talking. I would love to hear it from the patients who had the opportunity to have that drug, you know, based on that early result. Was that enough for them? If it wasn't,
number of shots on goal. Yeah,
that's right. I don't want to speak for patients in that regard, but I always believe let the patients decide. They're the ones who are the risk.
And that's exactly why these groups, the patient groups, are so important, is because it gives us the opportunity to know enough in order to be able to like push back a little bit, get some information. And I can't honestly think of a better way to end our conversation on that note, honestly. But if somebody has any just burning question, just speak now, or we'll have to circle back. Okay. All right, cool. No, I honestly just really cannot think of a better way to end the conversation than it's number of shots on goal. And, you know, you miss 100% of the shots you don't take. And so we need to be taking more shots. And I know that whether it's me or one of my family members, it would matter. I would like to have the opportunity to be able to do a phase 1 study if that's what I thought was the right thing for me. So, but thank you so much for being here and joining with us tonight and just answering the questions and being so candid and approachable. Your time is so very valuable, and you're always just a joy to chat with. And so, I'm hopeful that we'll be able to get you scheduled so you can come out where we're doing the Charlotte Connect event in here in September. And but I just really would like to thank you very much for being with us this evening, Dr. Kim.
Yeah, it's my pleasure to spend time with each of you. You're the brave ones. You're the ones who are helping drive it all. So I think that's great, and it's really appreciated, your advocacy, your bravery, and putting it out there. So it's our job just to help continue and support that. So thank you all.
Okay, well, thank you so much. Okay, guys. All right, well, I'm going to end it up for tonight, and I'll be talking to y'all real soon in Facebook group, and you'll be hearing back from me too. Okay. All right, guys. Well, thank you so much again, and I'm going to start the recording, and we'll talk to y'all soon.